neuroinflammation, and neurodegeneration for retinal vascular diseases, the major blinding conditions in the developing world. Most of these studies have an overarching component of translation from bench to bedside. With this, I invite Dr. Kaur to deliver a talk.
Thank you. Thank you. At the outset, I would like to thank the organizer and Abhijit for inviting me to present my work here. And I'm really glad because this week we are celebrating the safe prematurity. And yesterday was the ROP, retinopathy prematurity day. So on this auspicious week, I'm able to present this work that we've been doing on ROP. So as my title indicates, my focus today of my talk would be on the retinopathy prematurity. So retinopathy prematurity, as the name indicates, it's a complication, ocular complication of preterm birth, which is characterized by a presence of abnormal blood vessel growth in the eye, leading to the detachment of retina and ultimately blindness. And this is one of the major cause of childhood blindness, especially in developing and middle income and low income countries, as the graph indicates here. So about 20, like in India, and these are the statistics of globally with the percentage of preterm births and babies who develop some form of ROP, and then those who need some kind of medical intervention and eventually who go blind. But the current statistics in India, we have about 25 million preterm births, and of those, about 40 to 50% develop some form of ROP, and then 18% of these, they develop severe ROP and they go blind in just 1 year of age. So it's a huge problem that needs to be looked at. One of our institute, I'm from LV Prasad Eye Institute, Hyderabad, and one of the initiative that has been taken from our institute is on newborn health and also to minimize this blindness in pediatric population. And this is one of the annual report that I'm showing just to give you a glimpse of the activities that are being done, which is largely funded by Cognitive Foundation and Hyderabad Eye Research Foundation. So here you can see a clinician who goes to, so we have a network of ROP screening activity across three states in India, the Andhra Pradesh, Telangana and Odisha. And all the ophthalmologists who are participant in this program, they actually go to all the NICUs and screen the preterm born babies. And if they require any medical intervention, then they are treated either there or they are brought to our hospitals for treating these babies. So what is ROP? First, before I talk about my work, I'll just describe you briefly what is ROP. And for doing that, I'll just show you the typical eye structure for those who do not know. So this is our eye structure. This is the pupil, this is the lens, the outer covering here is cornea, and this white layer that you are seeing is the entire layer is the retina, which is then followed by retina is retinal pigment epithelium and the choroid, which supply nutrient to the retina. Retina is largely consists of neurons. So if a light falls on this, on the eye, the signal is perceived by the lens and the light is directed onto the retina. Once the neuron receive the light signal, they convert into the chemical signal, and these signals are then given to the brain, and that's how we see. So the functioning of retina is very important for us to be able to see. And losing this neurons at very early actually is very economically challenge and also a huge emotional challenge as well. So what happens in the eye vasculature, as I just mentioned, is very important for the neurons to function properly. And this vasculature formation starts at 16 weeks of gestation and it completes by 40 weeks of gestation. However, when a baby is born preterm, then the vascularization is not complete. That means the entire retina is not vascularized. And further to that, because these babies do not have well-formed lungs and other, these are put in the ventilators too and given oxygen supplementation. Now, once these babies are given oxygen supplementation, it actually suppresses the VEGF signaling, and and that causes the vaso-obliteration. That means the cessation of the vessel growth. And once these babies are out of NICUs, now because the blood vessel vasculature is not formed, they suddenly feel hypoxia, and by the time the neurons have also started to function. So that adds to the hypoxia, the energy requirement increases. And in that condition, the VEGF suddenly gets upregulated, and we see abnormal blood vessel formation, just like what we're seeing here. And that these blood vessels, they are very fragile, they leak, they form scars, and that causes the death of the neurons, and eventually the detachment of retina from its position, causing blindness. So this is just the classification of ROP. So if you see, this is a normal eye with fully developed vasculature, whereas when a baby is born preterm, this is the scenario. Half of the retina is avascular and half of the retina is vascularized. And slowly you see this position, this is the demarcation line that starts to develop, which is seen very clearly in stage 1 of ROP, and that gets deepened further, leading to the formation of ridge, which you can see from mild ROP, and in the stage 3 ROP, you can see that from this ridge you see fibrovascular proliferation. These are the fragile vessels which I was referring to, which leads to the leakage of blood and causing scar. And here you see the retina starts getting detached from its position, and finally here the complete retina is detached, leading to blindness. So there are a lot of challenges in the management of ROP. First, that we do not know which are the which of these preterm babies would actually develop ROP. So despite of lot of clinical research done on identifying the risk factors for ROP, other than gestational age, there is no other risk factor which can actually predict the severity or the development of ROP. So first, we need good imaging and biomarkers or other molecular-based biomarkers that can help us in predicting which are the babies who are at risk of developing ROP. Now, why this is important? It is important because there is typically no disease at birth. All the entire condition is about half of the retina not being vascularized. Now, this is a self-regressing condition, which means that the vascularization can complete normally, or even if there is a ridge formation, it can go back to the normal. So a clinician has to literally wait until the vascularization is completed before they can actually intervene. And so there is now some other adding problem to this is that means we need some kind of good diagnostic tools. Right now, the way we are diagnosing is based on only by looking at fundus of these infants and then see how they are progressing. And when you see a thick ridge formation, that's when you try to intervene. So we really need good biology-based markers which can help in early detection of this disease, and also which can help in monitoring the progression, that which are the ones who will progress to severe ROP. Further, we also need timely and suitable medical management. Right now, currently the management approaches are to burn these growing vessels by using laser or cryotherapy. And these don't help, then the vitreous is removed, because this vitreous is having the VEGF molecule, which this VEGF, to reduce the VEGF load, vitrectomy is done, and that is the second mode of management. And third is giving anti-VEGFs. Now, anti-VEGFs is a big challenge because these babies require VEGF for their normal neuronal maturation. So we can't really give too much anti-VEGFs, and then VEGFs are not a solution. It may be a recurring thing. So we need more better and stable solutions to manage these conditions. And also the screening tools that currently we have, which is more driven by professionals like ophthalmologists. So we need better tools which can help in screening these babies at the rural setups or in the communities where there is not good hospital available. So these are the challenges that we are working on to develop technologies based on both the clinical data that we have and also from the molecular biology data that we have generated in our lab. So now I will show you some of the data that we have done in quick summary of that, and then the technologies that we are trying to develop to address this problem. So in my lab, we are working towards using the omics tools to first identify and understand the genetic mechanisms and other molecular mechanisms that are involved in ROP development and progression. And for that, we are not just taking a single approach, we are looking at the prenatal aspects, postnatal factors, and further validating them by using some in vitro models, and then also taking, correlating our findings with the clinical phenotype that we get. And for doing so, we are using blood samples, saliva samples, placenta, fibrovascular membrane, vitreous humor. I'll explain these as I go for our experiments. So our initial genetic study, which was where we did a genome-wide association analysis with SNP arrays, indicated several genes in complement cascade, extracellular matrix remodeling, HIF1 signaling, and lipid metabolism to be significantly associated with ROP. Further, we also embarked on another program where we actually collected twin samples because our genetics in ROP itself was debatable. So to show that there is a genetic susceptibility for ROP, we actually worked on twin samples. So the twins where both the twins have ROP, where only one twin has ROP, another doesn't have. So discordant and concordant twins were taken for this analysis, and then we performed the whole exome sequencing to identify the genes. Now, and also the pathways. Parallelly, we also did the transcriptome, proteome, and metabolome profiling from the biological samples that we collected from the infants, and then correlated these studies together to really come up with the genetic pathways that are involved in ROP development. So from our transcriptome analysis, we found Wnt, immune inflammation, metabolism, MAP kinase, fatty acid metabolism, etc. are the major pathways that are associated with ROP. Now for doing the metabolomics and the proteomic study, we didn't use the blood samples, we actually used vitreous humor. Vitreous humor is a jelly-like substance present in the vitreal cavity lined by the retina. And it is believed that the insults or the changes in the retina can be seen in the vitreous, which is just adjacent to the retina in the eye. So these vitreous is collected at the time of vitrectomy surgery, and for control we use cataract babies who go for cataract surgery because there also a part of vitreous is removed. And so when we perform the proteomic study, again what we found that there is increased expression of complement components, particularly those which are involved in the alternate complement pathway, and this was confirmed by breast radiography. What was interesting to note was that there is complement present in the vitreous of these babies who do not have any ROP, who are just operated for cataract. However, in the cases, which is these babies who have ROP, you see multiple fragments of complement, and these correspond to the activated fragments that we call. So when a complement pathway is activated, there are proteolytic cleavage that leads to these fragments, and one of these fragments, particularly this one, is implicated in pathological angiogenesis. So we found this particular molecule only in cases and not in control. Further, we did mass spec analysis, and again in mass spec we could see increased expression of the peptides related to complement pathways. And interestingly, we also saw that while we see increased complement activation, we see a downregulation of anti-angiogenic molecules in the vitreous. And these are some of the molecules that we are shown interplay of glia and complement pathway genes in eye development and maturation, which I am not talking about here today. So another pathway that we found, from our genetic study was extracellular matrix regulation. And again, the proteomic studies further confirmed increased MMP secretion in the vitreous humor of the ROP patients as compared to control. So then we thought, what could be causing the synthesis of this MMP, whether it is RP cells or is it any other cells. So we did a cytoscreen analysis of vitreous specimen, and we found that there is the expression based on the expression, the presence of macrophage-like cells and neutrophils in this vitreous. Now this explains that probably these macrophage-like cells, the vitreous neutrophils, which are already known to involve in inflammation and lipid metabolism, are actually contributing to the MMP increase that we see and the complements that we see in the vitreous of these ROP infants. And this MMP is which secreted by the activated macrophage in response to ischemia or the oxidative stress degrades the basal membrane, and that causes a gap and leakage of blood and into the eye, which further compounds to the retinal detachment and further loss of vision. Now, one of the interesting findings that we had was when we thought this vitreous is having increased MMP, we characterized the fibrovascular membranes which are formed at the junction of vitreous and retina to see if there is increased expression of MMPs. And they did found increased expression of MMP9 in these fibrovascular membranes, but correspondingly we see a decrease in the opticin, which is one of the major constituent of vitreous. So that shows that opticin is a small leucine-rich glycoprotein, which is one of the substrate for MMP activity. So probably the increased MMP is leading to the downregulation or the degradation of opticin, and opticin is an anti-angiogenic molecule. So further studies that I'm not elaborating here, we actually showed in using a microglial cell culture model that actually, and if you inhibit MMP activity, you can rescue the opticin degradation, and that's how the drug that this can be one of the therapeutic targets that for preventing the progression of ROP. Parallelly, because we found these microglia, activated microglia in the vitreous, we also looked at role of these microglia, and for this we used a biometric scaffold which are designed into, they were randomly oriented fibers and we aligned fibers in scaffolds, and we grow the microglial cells on it. What we found that microglial cells that are grown on the aligned fibers, they actually are upon hypoxic exposure, they remain anti-inflammatory, whereas the ones which are grown on the randomly oriented fibers, they actually turn to become more pro-inflammatory. So that means these biomaterial scaffolds have a major role in shaping the phenotype of these cells, and this can be utilized for making the implants that we are trying to make for this disease. Next, we have shown you that the vitreous has excessive MMP9 and complement, but this was done only in the severe. Vitreous can be collected only from the severe stage of ROP. Now we wanted to know whether with MMP levels or which means inflammation in the eye can be tested right at the beginning before a child progresses to severe eye catecholamine. And then the only tissue that we can help use is the tears. So first we did a assay to check if tear can also show the similar level of inflammation as can be seen from the ROP eyes. And so for that we collected tears at the time of birth, at the time of first screening of these infants, and then we followed up these babies for until they develop or not develop ROP. And what we found here that tear MMP levels can reliably predict the progression of ROP. Because if you see here, these are the babies who developed mild ROP and who had regression of disease, whereas these are the ones who have the progression of disease. So those who have progression of disease or who go on to develop severe ROP, they had very high inflammation right from the beginning. So that means one of the MMP9 and complement C3 can really predict the inflammation in the neonate, and that can be used as a marker. And now we are actually, based on this, we submitted a grant to Inderjeet to develop a zinc oxide based sensors to measure the inflammation in these eyes, and so that this can be used for a community based screening. So I'm not going into this detail. This work we are doing in collaboration with Dr. Shiv at IIT Hyderabad. Next, we also did the metabolomics of vitreous humor, and these were some of the metabolites, one being the fatty acid metabolic pathway, which was severely strongly associated with ROP. Now, fatty acid, particularly the PUFAs, are the major component of the neuronal and the endothelial cells present in the retina and are important for maintaining the physiological functions of this. It is known that under oxidative stress, these PUFAs are oxidatively modified, and they cause the imbalance in the retinal homeostasis. So I'm sorry, I'll just skipping this slide. There are few other studies where people have shown that in the serum they see in low fatty acid levels, and that was linked to ROP pathogenesis. So what we did, we worked on this omega-3 fatty acids, particularly arachidonic acid, and looked at different enzymes that are involved in this metabolizing this arachidonic acid into this metabolite. And three major pathways there are. The one is the FOX pathway, lipoxygenase pathway, and the CYPs, which are involved in the metabolism of arachidonic acid. Now, we found there is increased expression of FOX, LOX, and CYPs, which leads generating the inflammatory metabolites. And further, these metabolites were acted upon by the soluble epoxide hydrolase, which converts this eicosanotetroic acid into dihydroxyeicosanotetroic acid. And this enzyme is actually reduced in this concentration in the expression. Now, this soluble epoxide hydroxylase, actually, if it is reduced, it means that there will be reduced expression or the formation of DHETs, which are important for the astrocyte functioning and leading to astrocyte loss. And these astrocytes are again very important in retina for forming the proper neovascular growth. Further, these DHETs are also involved in causing abnormal angiogenesis by a Notch signaling pathway. So we measure these different molecules which are present in the signaling, and we show that even in the absence of epoxide hydrolase, there is reduction in the level of Presbyopia 1, and also the increased expression of Notch, which causes upregulation of VEGF and apoptotic marker like caspase-3 and caspase-1. Again, when we looked at this particular enzyme in placenta samples, their expression was reduced in the placenta for preterm who developed ROP. Further, as I said, that these metabolic pathways generate a lot of inflammatory molecules, and this corresponds to our previous data from gene expression, where we showed these pathways NF-kappa signaling, which actually again causes increase in the inflammation and also the cell death by various pathways. And so what we propose that there is compromised immune system and upregulation of arachidonic acid metabolism related enzymes, and this is associated with inflammation that is seen in this ROP infants. So these are just a summary of all the pathways that we identified from different approaches, and this is our current program that we are doing, where we are actually typing the mRNAs and also doing the more in-depth analysis of the molecule, and also coming up with a dual-loaded drug implants to treat inflammation and neovascularization, which can help in combating ROP. So this is the plan for taking this research forward, and this is the entire team with whom I work, and this is the grant funding support that we have received. I thank all the patients and the funding agencies for providing support to this research. Thank you so much for your kind attention.
Thank you, Prasad. industry as well as the grant agencies. We will be able to see some translation sometime soon. I really want that should happen.
Hi Inderjeet, a lot of data exciting talks. So my question and comment is more on your angiogenic data.
Right.
So since it is happening in the kids, right? So there is no point of inhibiting angiogenesis because that is crucial. What I noticed in one of those experiments which I did at ICER, this doesn't belong to Biocon, that when you put RPE cells on soft versus stiff substrate, and Abhijit might appreciate that, so VEGF secretion automatically goes up when cells are RPE cells on stiffer substrate. And then instead of targeting, of course VEGF is important, but instead of targeting that, can we target some of the mechanical factors? Can we target some of the proteins which sort of regulate stiffness of vitreous humor? Did you check how stiff is vitreous humor in natural, like normal versus disease kids? Thank you.
Yeah, so we haven't really done it, but this thought is definitely there, and that's why I said, targeting VEGF is not really a good solution, especially in a developing child. So we are also looking at means to target other molecules, and that's why we have been doing these omics-based studies to identify the molecules which are upstream of VEGF signaling, which can be used for drug targets. Yeah. So that's why my approach right now is to look for the inflammation, which because that is indirect, contributing to angiogenesis. But if we can control the inflammation, in fact, I just want to tell you that when we did these vitreous biopsies and cytospin, we found these inflamed cells also, like neutrophils and other. And then my clinician told me that long back when ROP was characterized, in fact, media haze was one of the, media haze means presence of inflammatory cells in the vitreous, is one of the criteria which was noted down, but in the regular practice nobody looks for it. And after looking at this data, she started looking and she said that this can be one of the markers which can help clinically for understanding or monitoring the progression. However, coming back to your question that vitreous hydro, we are not working on vitreous, but this hydrogel aspect, that what I showed you with what I'm working with IIT Hyderabad, that's what we wanted to look at, whether these kind of biometric scaffolds can help in somehow combating this inflammation, which is what we are seeing is contributed by macrophages and microglia. That's what we are doing. But definitely look forward to your
And so long just to bring this mechanics into the picture, we had
hello. Yeah. So, ma'am, it was a really good talk. So, were you able to correlate ROP with any sort of a bacterial infection? Because bacterial infection is a very big cause of preterm births.
Right.
So, have you done any correlation with that?
In fact, not just bacteria. The major commonest cause of premature birth is actually Candida in India. And we have looked for both the Candida and bacterial infection. So sepsis is one of the common conditions in premature, but the samples that we have collected are from the infants who did not have event of sepsis, because that would then confound, that will act as a confounder. So these samples were really those who did not develop any sepsis or any bacterial infection.
And one more, that you saw that there was high amount of inflammation in placenta.
Right.
So, ma'am, what was the cause, and also placental function is to kind of prevent that inflammation to go across the barrier and reach the fetus and protect it from anything of such sort. So, if that is a cause, so what was the major cause for development of
actually,
infection?
I didn't show the entire data. We actually did a comprehensive analysis of this placenta. We found very high HIS1 signaling in this placenta. And also histologically, we have seen that things like chorioangiosis or the high ischemic changes in this placenta. So I think this is contributed by oxidative stress, but what causes oxidative stress is something that we need to look at.
Hello, ma'am. Ma'am, actually, I have a very naive query. There are some therapies, obviously in literature only, for preeclampsia that supports angiogenesis in placenta. So can those therapy also help in progression of RP?
I have no answer for that because I don't know much about it, about these therapies. But that's what I'm saying. So it's not about vascularization. Vascularization is important, but what we are talking in what we see in ROP is the abnormal vessel growth, which is like thin vessels. So if there is a approach which is only supporting a normal vascularization, I don't think so that should give any harm. What we need is to curb this, aberrant vascularization of cells, which is seen in
Okay.
Ma'am, it was an interesting conversation, the slide what you showed. Firstly, I would want to know the duration of the therapy, what could be given for the children. Secondly, you focused on the inflammation which basically cause ROP. Could there be another causative factors which could lead to ROP? And that's all.
Okay, so pardon me if I forgot any of your points. So you made 2 points. One was that what is the duration of therapy? So the course of therapy is like that when a ridge deepens up and we see fibrovascular proliferation from this ridge coming up. The first thing that is done is laser therapy or cryotherapy to burn those vessels which are coming up. And if that helps, then that stays there. But most of the time that doesn't really help. It only gives temporary relief. And then if we see further these vessel formation, then the next course is to either do vitrectomies to prevent the retina from detachment and to give anti-VEGFs. So anti-VEGFs have been started very recently because there are a lot of concerns about using anti-VEGF. So duration typically is from maybe 2 months to 1 year. And after that, the course is normally, there's nothing much that we can do if the eye gets into detachment. So then only anatomical correction can be done, which can save some vision, but not really. It can't bring back the vision. Now the second question that you said about inflammation, yes, we first identified inflammation as one of the key event that is happening, but we also are looking at the other aspect which I said that which is being worked on is also on the neurodegeneration and also like lipid metabolism. So we are working on it. There are other indicators also. One of that is also that if say for example there were from other groups they have shown what he just asked about sepsis or ocular infection. We haven't seen infection in these cohort that we have analyzed for our study.
Yeah. So you said this there is an elevation of MMP2 and MMP9 causing this inflammation. Now this amniotic fluid and amniotic membrane, they have TIMs.
Yeah.
So have you done any trial where using of amniotic fluid or amniotic membrane can bring down these MMPs? Any in vitro data, anything?
No, we haven't done. We just measured these MMPs in the placenta.
Okay.
Particularly the amnion-chorion interface, and we see increased inflammation even there. And also I've shown you the complement data and the foraminifera. But we haven't tried to look at it. Thank you.
Thank you, Dr. Kaur, for that lovely talk. Requesting Abhijit to hand over the memento to Dr. Kaur. Right, so we will now go into our coffee break. This will be a little extended than the time given in the schedule, so we request you all to be back in the auditorium.